Good morning again, and thank you for being here. So it's the first time to give this talk from me. So it's all about immunology, and I am a genome scientist. So let's see how it goes. But we were interested in, because we met a professor Hirose a few years ago, and he had a very interesting, very important collection of supercentenarians. So what are the supercentenarians? Supercentenarians are guys that live 110 years or longer and spend their life until after 100 years old. They are totally healthy. They have no disease, no impairment of knowledge, anything. So they seem to be really live a very to have a very excellent life. And we were wondering what makes them. I think it's okay. I think it's okay. We were wondering what makes them so. One of the problems in this study is that the supercentenarians are not so many. The calculations are up to 147, 149 when they count them. And not all of them agreed to participate in the study, so you can count only on very few of them, and you cannot follow up them for many years because, unfortunately, they are just at the end. So this is a little bit how the study is. So samples are very precious, and we were wondering what we can do with some of the samples. We can sample only 2 milliliters of blood at maximum, and then after you negotiate with the family, so and then you really is really very precious sample. So we were wondering what we can do, and because they live they are very healthy for most of their life. What we try to see at the beginning, if we can profile their immune cells, and so we started with the PBMC. And at that time, the 5' approach was not ready yet. We didn't have enough data, so we decided to be conservative. Take the whole blood for 7 supercentenarians and 5 controls. Take the PBMC and do the 10x 3' sequencing and count the expression of gene. In the presentation, SC stands for supercentenarians, and CT are the controls. Does it work? Yes, we can provide those cells, so we have in total about 61,000 cells from 12 donors, which 7 are the supercentenarians. And by Sunday for Sunday check, we've been checking all the markers and essentially. It makes sense by looking at the data. We started to analyze a little bit more in details, particularly the class TC1 and TC2, and annotated them based on the marker genes. And in particular, if you look at the supercentenarians, actually we see that. In the supercentenarians, the class TC2 is much higher than in the controls. And what is happening here is actually we've been analyzing the expansion of cytotoxic T cells in the supercentenarians, and actually we have been analyzing, and actually, if you compare to the supercentenarian to the control, so we found that the class the TC1 is much higher in the controls, and the TC2 class is much higher in all of the supercentenarian samples. By further analysis of gene expression by several of the TC2 cells that are again expanded in the supercentenarian, we started to identify genes that are associated with cytotoxicity. So essentially, granzyme H, B, A, and the PRF1 that are essentially associated with these clusters, and by further analysis, so we found that them associated to essentially over-represented in all the supercentenarians, and if we compare to the controls samples. And again, so some more data here, and the key point is that in the in the supercentenarians, again we found that the number of genes identified per cells were very much over-represented. So essentially, cytotoxic T cells are expanded in the supercentenarian. This is a summary of all the cytotoxic T cells, if compared to the controls, and we further analyze a little bit more in details to analyze what is in the CD4, what is in the what are the CD4, what are the CDA and TCR delta, and actually we found that there were many unexpected, many CD4 cytotoxic cells, and in particular, so it was very clearly, if you calculate with this class, so particularly the CD4 that usually are not cytotoxic, they are very much expanded in the supercentenarians if compared to the controls. This is quite striking. So I am the CT1 actually, so I'm very different from the supercentenarian. So I'm not sure if it is good or bad, but and the CD8, of course, they are also expanded, but not as much as a CD4. So the key point is that this cluster is very rare, but is very high in the supercentenarian, where usually is no more than 2% in normal samples. I will skip this, and again some recounting of the number of to show that statistically is relevant. So essentially, the CD4 granzyme positive are expanded in this class, and then we wanted to see if there is any special clonal expansion, or if the whole class is expanded. So we've been looking at the T cell receptor, because we can sequence, we could resequence from a couple of supercentenarian, and then by sequencing the T cell receptor, we've been analyzing to see if they are clonal, or if there is a large variety of them. And actually, we could call back the supercentenarian we call number 1 and number 2, and we've been sequencing the T cell receptors and the transcriptome. And actually, what happens is that by analyzing either helper cells or CD4, we have identified that while the helper cells in the supercentenarian 1 and the supercentenarian 2 tend to correspond to a large number of cells, for the CD4, actually found that very few cells have been expanded to a large number. So in the donor 1, 69 became 908, and the 325 cells became 2011 cells in our analysis. In particular, if you look in more details, we found that particularly some of the cells have been massively expanded to occupy up to 45%, 50% of the whole repertoire of the CD4 cytotoxic cells. And so essentially few of them have been expanded. But of course we don't know yet what are they for? So we'll still, it still will take some time to identify if they are specialized against against any specific antigen, against any specific response or not. The final question is that are those cells really cytotoxic or just express genes? And we have been assaying by stimulating them with PMA and blefeldin, and actually by further analysis we identified that they express interferon gamma if compared to the control, and they also express TNF alpha. So essentially, it seems that their cytotoxic activity is very likely to be real. So as a summary, the key point is that the helper CD4 T cells are differentiated into cytotoxic cells, and they are clonally expanded, and they are actually active, and we don't know the specificity of the antigen yet. The paper is going to be out in PRONAS tomorrow, in 22 hours, so please have all the details. It was everything by archive. To thank many collaborators, particularly Hirose Sensei, who has been collecting all the supercentenarians, and Koso Kashimoto for organizing a lot of this. Thank you for your attention.